just give your undivided attention to Dr. Ludwig, please, and we're talking about EEG and the experiment that many of you participated in. Go ahead and take it away.
All right. You have talked about EEG in this class already, right? About how it's done. So I'm going to go through a quick review of, whoops, might help if I plug this in. Too many things happening at once. Let's try again. That's better. Okay. So the EEG signal, electroencephalography, uses electrodes right on the scalp, and it records the very, very tiny voltage changes that are obtainable at the surface of the scalp. So we have to recognize right off the bat that we're not measuring an individual neuron's activity. We're not even measuring a group of neurons. We're measuring the combined activity of millions of neurons, and therefore it gets a little tricky interpreting exactly what that activity shows. But it gets more useful when we compare one part of the brain with another part of the brain. Let's say an electrode placed on the left hemisphere versus an electrode placed on the right hemisphere, and looking at the pattern of activity in a global or gross way on the top of the scalp. Now, raw EEG has some clinical uses. For example, if we're trying to diagnose medical conditions such as epilepsy or brain damage, we can just look at the raw EEG and draw some conclusions from that. Raw EEG is also used in sleep research and to a lesser extent in some types of cognitive activities, such as the overall activity of the left hemisphere compared to the overall activity of the right hemisphere. But here's an example of how it could be used to diagnose an epileptic seizure. Because here we have a group of electrodes, like 16 electrodes, and before the seizure happens, they're minding their own business, showing kind of normal variations, and suddenly we have this massive synchronized wave pattern breaking in over a period of 45 seconds or a minute, and then returns to a lower level of activity. So this is a pretty clear sign of one of two things. Either something's gone wrong with the computer, or the electrodes, or the person's having an epileptic seizure. Okay, somewhere along the line you learned that EEG comes in different bandwidths, and that can be filtered out, looked at specifically. When you're awake, you're typically giving off the main EEG signal in the range of what we call beta or alpha. If you're falling asleep, you're giving off theta waves, and then if you're deeply asleep, your EEG is going to show mostly delta. There's also this very high frequency waveform called gamma. We don't really understand what it means yet, but there are a lot of people who think that it has something to do with connecting the activity of different regions in the brain, and therefore it might be important for conscious awareness. So if you hear a sound and you see something moving, the fact that your vision and hearing can be connected so that you come to believe that that moving object is generating the sound, then that would be an example. I mean, since the sound and the sight are being processed in different regions of the brain, somehow we need to connect that information together, and it could be gamma waves play a role in that. Okay, but alpha waves, although they're legitimate waves that occur when a person is awake, cause a problem in EEG research for two reasons. One is they're massive compared to the beta waves, and so they kind of wash out a lot of the other activity. The second problem is that they usually indicate the person has lost focus. And so if you are trying to pay attention to the screen and you're sort of drifting off and not noticing what's going on anymore, your brain's going to be producing alpha waves, and so this is a bad sign. And so the software that we use will throw out a trial if there is evidence of alpha wave activity during the key portion of that trial. We don't want that. We want focused attention on the cognitive task. You need to be paying attention to what we ask you to pay attention, or else the brainwave is not going to tell us what we want it to tell us. Okay, so remember that the raw EEG is picking up all of the neural events that are happening at that moment in time in that region of the brain. And that means there's a very high signal, very low signal to noise ratio, a lot of noise. Because while the thing is happening on the screen and we're trying to get you to pay attention to that, your brain is also doing other things. It's processing sounds and the pressure of the chair against your back and all that sort of stuff, and might even be processing your thoughts about what you're going to do this weekend and so on. And so all of that random kind of activity is interfering. So what we need to do is come up with some way of reducing that background noise. And that's why we move to ERP, or event-related potentials. So rather than just looking at raw EEG, what we're going to do is take slices of that raw EEG where we, the experimenters, have somehow controlled the situation, calling your attention to a specific thing that happens again and again and again. Like we can play a tone of a certain frequency again and again and again, and watch what your brain does in response to that specific tone, and filter out all the other things that you were thinking about before and after that tone occurred. Does that make sense? And so ERPs, event-related potentials, have a particular profile. And so over time, researchers have identified what we call components. And so this is called the P1 component, this is called the N1 component, and they're named in relation to whether the deflection from the midpoint here goes in a positive or negative direction. And due to a historical quirk, often the ERP plot is upside down compared to what you would expect, so that many researchers plot positive deflections going down. So notice it's really important to look at the scale on the vertical axis of an ERP plot, because frequently you will find that the positive side is down and that something that looks like a positive spike is actually a negative deflection. Got that? So here we have P1, P2, and P3. P3 has been studied a lot in terms of cognitive processes. It seems to have something to do with memory updating or noticing that something has changed in a visual display. And then the negative components haven't been studied as much, but one that I'm going to talk about occurs at about 400 milliseconds after the onset of the target, whatever it is, a flash of light, a sound, whatever. In our experiment, it was a language cue, a word was presented, and the presentation of the word is considered 0 on this scale. And then we're looking at how your brain reacts in the moments after that target was displayed. So we measure two things in an ERP experiment. We're measuring the amplitude, how much the deflection goes away from the center point, and we're also measuring the latency. How long after the target was presented do we see this peak or this component, either in a positive or negative way? So the early peaks before 100 milliseconds after the onset of the stimulus, not much cognitive processing has gone on. It's mainly just the sensory reaction. In vision, we'd see this occurring primarily at the occipital lobe as the visual cortex is reacting to the light that was flashed. If it's an auditory experiment, we'd see these early things most strongly in the temporal lobes near the auditory cortex processing sounds. But the further away you get from that 100 marker, the more cognitive the thing appears. It's no longer just a kind of a knee-jerk reflex to the event that was presented. It's how your brain is thinking about that and relating it to memory and relating and trying to understand the meaning of that event. So that brings us to the N400. So once again, I don't have it on this thing. Here we see a P300, which, why do we call it P3 if it's over the 400? Because you can see the deflection starting at around 300, and then often it reaches its greatest deflection at about 350 or 400, but we still refer to it as the P300. By the way, P3 and P300 are basically the same thing. it's interesting that the time scale as to when these things happen, like the P2 is the second positive peak, but it often occurs at about 200 milliseconds after. And so if we're talking P1, P2, P3, we're just counting. If we're talking P300, we're talking about the latency after the onset of the target. But often they line up. So P3, P300 means the same thing. N4, N400 turns out in most cases to be the same thing. Okay, so this is an actual plot from our lab, our EEG lab, showing, some of the charts I'm going to show you have negative up and some have negative down. Don't be confused. Just bear with me. Realize that it's arbitrary which way we flip this. Okay, so here's the P300. Here's the N4 or N400. And what I want, the reason I show you this slide is to get some indication of the variability across trials. This is the same person over many different trials, and you can see that this particular peak was really strong on one particular trial, but not as strong on the others. And so it's helpful to average across all the trials so we get a clearer idea of what's happening. And so here you see another way of averaging. In this particular experiment, one of the initial experiments that demonstrated the existence of the N400 component, they had, I think, 12 or 16, they had 12 subjects. And so the left part of this graph is the waveform, the average waveform of all the trials for one subject. And then we plot that for each of the 12 subjects, and it's kind of a mess. But then if you do the grand average across all 12, the average across all the trials for each subject, and then average all the subjects, you get something that really shows the N400 as a distinctive waveform component. Okay, in our demonstration, put up your hand if you actually participated in this. Okay, about half of you did. You went through 160 trials. Each trial there was a 7-word sentence. Half of the sentences ended with an expected word that made sense in that context, such as, he ordered pizza with pepperoni and mushrooms. That makes sense. Maybe you don't like mushrooms, but at least you understand that the sentence makes sense. Half the sentences were matched, but they ended with a word which made the sentence rather weird. He ordered pizza with pepperoni and socks. When your brain hears that, it doesn't like it. It's trying to make sense of it, and it doesn't make sense, and so your brain's doing something different with that particular sentence. Okay, as those of you just saw or heard me talk about in the tour, the first 6 words were presented, and then there was a pause, and the last word was presented. That last word made it either a normal sentence or a nonsense sentence, and so that's where the trigger was sent to the EEG computer so that we can do this time-locked ERP analysis to the presentation of the last word of the sentence. So the purpose of this demonstration experiment was to try to replicate the original experiment having to do with the presence of the N400 on nonsense trials. And so what the participant experienced was to be fitted with a cap and then have the electrodes placed in the cap. Here are 64 positions on the scalp. We used only 32 of those positions. The ones I'm going to talk about primarily are the CZ, the FZ, and the PZ, because it's just, those are convenient reference points. But because this is a language experiment, we typically get more activity on the left side of the brain than the right side of the brain. And so when I've looked at which of these electrodes is actually showing the N400 most strongly, it tends to either be this, for some people, this CP3 or C3 or FC3. This is kind of over the left temporal area near the auditory area and also what we would call Wernicke's area, which is responsible for the interpretation of spoken language or written language. Okay, so I'm going to show you down the center line, but just realize that if we had picked the electrodes on the left side, we might get an even stronger effect. Okay, here are some photos of the person being fitted with the cap, electrodes put on. Why do you think we need these facial electrodes in addition to the EEG electrodes? Because people blink and they move their face. And moving the muscles of the eye or the face creates a tremendous artifact which can wash out the weaker activity that's being picked up with these brain electrodes. And so we have a manual way of removing the worst eye blinks if they occur during a trial, but the computer software also filters out a lot of more minor movements, such as moving the head or swallowing, those artifacts. And some of you, we lost some of your trials because you made a larger kind of facial movement that the software could not filter out, and so the trial had to be thrown out. Okay, I pointed out that we use a battery for electrical isolation, so none of the participants are in physical danger of having a shock to their brain. We have the two computers, one that presents the stimulus and the other that records the raw EEG, but the computer that controls the experiment and presents the stimulus sends a signal to the EEG computer at the moment that the critical word was displayed. That becomes moment 0 in our time-locked ERP analysis. Okay, so you've seen all this stuff. I've talked about this already. And so here's a screenshot of the actual recording on the EEG computer. And you can see that each of these 32 electrodes is producing its own kind of unique pattern. But what we're going to do is slice out from the moment that the stimulus is presented, the keyword is presented, and then 700 milliseconds later, average over all of those trials to get something. I mean, if you look at raw EEG, unless you're some kind of super genius that has a brain focused for this, it really makes very little sense until you do the filtering and averaging. And then you get something that looks like this, and you can start making sense of it. Notice what I've done here. I've flipped the scale. And so now we're talking about, if you look over here, these are positive numbers and these are negative numbers. And so here is the moment of truth, the 0 point. Here's 200 milliseconds later, so this would be N2. Here's 400 milliseconds later. And so all this downward deflection is actually starting at about 300 or 350, but in most cases it's most noticeable around 400, so we call it N4 or N400. Now, there's another quirk to this, and that is that there is a positive component that occurs at around 500 or 600 milliseconds after the onset of the trial, of the critical word, which seems to have to do with checking the grammar. And so if we have meaningful sentences that have grammatical errors in them, we tend not to get an N400, but we get a strong P600 that indicates that something clanged in your brain as you processed the grammar of that sentence. That sentence was not grammatically correct. In our study, we actually got a little bit of P600 activity along with the N400. So mainly we were trying to manipulate the meaningfulness of the sentence, but some part of your brain, which also is checking the grammar, didn't like the way that sentence ended. It kind of threw up a red flag in the grammar processor for your brain as well. Okay, as I pointed out in the lab, the black line always represents the normal sentence, the correct sentence. This is the average of 80 trials of normal sentences. The pink or red line represents the nonsense sentences, the average of 80 trials of nonsense sentences. Okay, so here we have, this is spring 17, and so I've got the data from the people who were actually participating over the past 2 months. And so you don't know what your number was, and no one else does, so you don't need to be embarrassed. But as I looked at them, we got some really strong examples of N400 here. Can you see this one? This is participant 17101, and you can see at 400 milliseconds, you can't read that, but I can read 100, 200, 300, 400. What we're seeing is a pretty strong pink line deflecting below the black line. So this person's brain was reacting very differently to the nonsense sentences than to the others. Here's 103, another good N400. 106, another good N400. This person, 110, showed a little bit of a N400 even on the normal sentences, which is not that unusual, but the difference between the normal and the nonsense is pretty clear. And here we have another example. That's a really good example of the N400. Here we have N400 with normal and nonsense, but the nonsense one is much stronger. Okay, then we had some people who didn't show a strong effect, but I would still say on balance that this reflects, this one's pretty clear. This one is like a P400 in the normal sentences and a little N400 with the nonsense ones. These two are a little less obvious, but you can, if you look carefully, you can see that the pink line is having a negative deflection that's greater than the black line. But then 3 of you, for reasons that aren't always clear, did not show the expected pattern. So this subject 104 actually showed more of a negative deflection on the normal sentences than the nonsense sentences, and the same is true of subject 115. And the person who was 113 either has a very fast brain, and so we're picking that up at 1 to 300 instead of 400, or was doing something completely different. Now, before I cast aspersions on any of your brains, we have to recognize there's always the possibility of experimenter error. Because when my research assistants did this statistical analysis of this, what if they flipped the codes for the nonsense and normal sentences so that the nonsense sentence turned out to be plotted in black, in which case these two would clearly be showing an N400 if they had made that mistake. But I don't want to say that they made the mistake, they probably didn't, and so I'm going to say that this is just variation. Not all brains work in the same way. Each brain is unique, and some people do not show the kind of average performance. That's why that Kudus and Hilliard article from 1980 was very careful to take the 12 subjects and do a grand average of all 12 subjects before they talked about the existence of the N400. If we averaged all 14 of our subjects, we'd have a really good N400. But that means it's an average, and not every student, not every participant showed the same thing. Now before I quit, I need to point out, nobody in this group of 14 participants had the problems that we had last semester. Last semester, we had one student who looked brain dead. When we analyzed none of the normal components were showing up. And so my conclusion would be either the person fell asleep or was not paying attention to was thinking about something else and pressing buttons at random, or there was something wrong with the electrodes. It just wasn't picking it up. And this is not participant error. This is electrical interference. Something was going wrong. And it could happen, the experimenters were pretty good at noticing if you were wearing jewelry or anything else. But let's say you had massive work done in your teeth or whatever. You had a lot of metal in your head some way. You were chewing on something that was metal. You had your iPhone in your pocket and you didn't give it up when you were supposed to. Some kind of electrical activity was messing this up. Didn't happen in this semester, but I just wanted you to show that sometimes we can't even use the person's data because the activity does not match anything that we know. Just quickly before I quit, let me show you the results from last semester where we did the same experiment that we did here. Once again, and here, what I'm showing is the 3 positions, F, C, C, Z, P, Z, for each one, because I wanted you to see that this particular participant showed a pretty clear N400 at the position CZ, center, PZ, more posterior in the parietal lobe, but not really in the frontal lobe area. And the next 2 showed it in all 3, These next two showed it in all three, but here's a person, once again, who showed it pretty well in the CZ and PZ, but not in the FC area. We had more participants last semester that only showed a moderate difference between the nonsense and the actual meaningful sentences. We had some people that just barely showed a difference, probably not even statistically significant. And then we had 3 people who didn't show any clear N400 at all. Okay, thanks for your patience. I hope that you got something out of this and that some of you will be motivated to continue on with EEG research in some way in the future. Do you have any questions before I leave?